So I am going to give you a little breakneck overview of how the prion idea is being applied in other neurodegenerative diseases. I'm basically going to concentrate on the ideas. I'll show you some scientific exploitation of the ideas, but I hope that I leave the podium with you, bubbling away about this new way of thinking about non-prion neurodegenerative diseases. This is the Prion Hypothesis, about as simply as it can possibly be reduced. You've seen some beautiful renderings of the normal cellular prion protein. What I've done is to make it look like a bed spring because it is rich in alpha helical content, what a structural biologist would call alpha helical content, all those curlicues. The abnormal prion protein, the disease misfolded prion protein, obviously has lost that structure on the left and formed another structure, which is rich in beta sheet content, hopefully conveyed by the saw teeth. So this is, as they say, this isn't rocket science, but really what makes this an epic setting idea is the fact that it's not just PRPC turning to PRPSC, and that's the end of the process. PRPSC then acquires the ability to convert PRPC, the normal cellular protein, into more of the disease-associated form. So a good guy and a bad guy enter the scene, and two bad guys leave. So that's basically the prion conversion in a nutshell. So I'm also going to basically take you back to high school. As you heard from Dr. Collins, and as I think we all learned in high school, a protein is actually a string of amino acids. It's a chain of amino acids, which is encoded by the DNA. But a protein doesn't acquire its function until it gets folded up into a proper three-dimensional shape. An example would be the shape in the middle. And this specific three-dimensional shape also enables the protein to carry out its function, whether it's an enzyme or a receptor or a pore in the cell or a structural protein or whatever. It doesn't work when it's totally unfolded. It must be carefully folded into a three-dimensional shape. But there are when a protein wears out, when it becomes oxidized, when it becomes fragmented, it gets converted back into its constituent amino acids by the degradation machinery, and that used to be the story with the life cycle of proteins until the Prion hypothesis was essentially formulated by Stan Prusner, in that a normal-shaped protein can become distorted, and this is a catalytic process. It keeps the dominoes keep falling. So proteins get converted to an abnormal, usually beta sheet-rich form. But we now know that you see I put there is, okay. We now know that this prion idea applies to other neurodegenerative diseases like Alzheimer's, Parkinson's, ALS. And I will show you some of the evidence for that. In my lab, what we've kind of been doing for the last 20 years is trying to figure out what parts of a protein are exposed when the protein is misfolded in disease. And why is that important? If something goes from the inside of the protein to the outside or the surface, it then can be a target for an antibody, a specific target for a specific antibody that will only bind to the sick protein and spare the normal protein. There are many reasons why that would be linked to efficacy if you have such an antibody or such a vaccine, and reasons why it would promote safety because it's not reacting against the normal protein, the normally shaped protein, notwithstanding what Professor Collins just told us about. So let's turn and switch to Alzheimer's disease, and I could ask you in this room if anyone has not been affected by Alzheimer's, had a relative or a friend or a parent, and the answer would be no. We all have, but I could do the same talk in front of a group of taxi drivers, and they would all say, yes, I've been affected directly by Alzheimer's. Alzheimer's is not only very common, I have the statistics here for Canadians and Americans and worldwide statistics. It is a disaster for an individual, a disaster for a family, a disaster for the healthcare system, and now we are learning a disaster for the health economy. It has been estimated that Alzheimer's in the United States, Alzheimer's care is on the order of $200 billion a year. So another statistic which is just kind of arresting is that if we were able to delay the onset of Alzheimer's, just delay it, not stop it in its tracks, by 5 years, that would save $50 billion in the United States. So this is a very important thing to go after, not only because of the suffering of individuals and families, but because we're going to be bankrupt as we get older and the disease becomes more common. So, can I talk about Alzheimer's being a prion disease? Come on, get off it. It turns out that there's been careful work going back decades, really, that are consistent with the prion idea. And here is some work from a neuropathologist named Heiko Brock, and what he did was postulate that Alzheimer's starts in one small region of the brain and then spreads throughout the entire cortex in a systematic fashion. This idea that Alzheimer's could have a prion-like component got experimental footing in the work of Larry Walker and Matthias Euchre, where they actually injected mice with homogenates from brains of people who died of Alzheimer's disease, and the mice that were special transgenic mice actually came down with a spreading amyloidosis that looked for all the world like Alzheimer's plaques. So this is something that can be transmitted from human to mouse. It can be transmitted in other animals. And there is a systematic spread of both the injected material and the pathology in tau, which turns out to also be a prion-like protein in Alzheimer's. So we have concentrated for various reasons on a form of A beta, this little peptide, which is toxic, which is called A beta oligomers. These are small clumps of A beta monomers. And this is because the seeding principle in the experiment that I just showed you is enriched in oligomers. Also, oligomers are thought to be the most toxic form of A beta in an Alzheimer's brain. So we were able to recognize and raise antibodies against an oligomer-specific epitope. And I won't go into the details here, but this is the shape of the oligomer-specific epitope. Antibodies against that do not recognize Alzheimer's plaques, so they don't recognize fibrils. They also don't recognize the other form of A beta, which is monomers. Oligomers are very present in affected regions of the brain in Alzheimer's disease, but not normal controls. And our antibody protects neurons from cell death induced by oligomers. So I want to spend a few minutes talking about amyotrophic lateral sclerosis. I've directed ALS clinics for over 30 years now, and this is the disease. It's a progressive paralyzing disease. In case none of you know this, I bet a lot of you do. But it's also characterized by intact mentation. So you're watching your own disintegration. And it's, the hate and the fear I feel with this disease is hard to articulate. This is a much less common disease, but it's very costly, as you can imagine. Healthcare systems are not going to be bankrupt because of it, but each individual case costs a fortune in medical care. So is ALS a prion disease? And this basically came out of hypothesis from observing my patients. So you get weak in your right index finger or your left big toe, and there'd be this systematic spread of paralysis through the nervous system. And we ultimately determined that there is a prion protein or a prion-like protein in ALS, which is called superoxide dismutase 1. I'll just show you a very brief experiment. We can we can insert mutated SOD1 in cells in culture, take the supernatant, layer it over fresh cells, and we can transmit the misfolding of SOD1. This can also be blocked with antibodies. I won't show you this information, specific antibodies directed against misfolded SOD1. And over the past few years, there has been a number of wonderful experiments coming from the Dave Borchelt lab at University of Florida, showing a mouse model in which SOD1 misfolding can be transmitted and induces disease in a particular mouse. So in our own lab, we find that giving mice one of these misfolding-specific antibodies prolongs their survival. It doesn't arrest the disease, but it significantly prolongs survival by about 25%. So I want to kind of sum up by saying that, as horrible as it is, the prion diseases, including Creutzfeld-Jakob disease, has provided us with a template for understanding neurodegeneration and for targeting specific components of this amplification pathway that recruits normal protein into a bad protein, a bad toxic protein. And I was going to show this, just a silly little cartoon showing that cells can infect each other. Neighboring cells can be infected by misfolded proteins, and that ultimately kills both cells. But the vision for an antibody or a vaccine is that this transmission is arrested and the nerve cell is protected. In other words, in the best case scenario, we might be able to actually arrest diseases like Alzheimer's, Parkinson's, and ALS. So I thank you very much for your attention. I did want to mention here that I founded this company, Promise Neurosciences, which also has the licenses from University of British Columbia to commercially develop these discoveries. So I thank you for your attention.
Thank you, Neil. We have a time, few minutes for a few questions.
On the previous slide that you had diabetes on there? Is that?
Yes, in fact.
That was shocking.
Yeah. So the prion idea has been, I didn't really have enough time to linger on this, but there is a type of amyloid that accumulates in type 2 diabetes, which is made up of a peptide called amylin, or IAPP. And this can be transmitted to naive mice. In other words, you can induce a type 2 diabetes like disease with fibrils from that are recombinantly or made out of bacteria. I think that Claudio did those first experiments. Claudio Soto did that work. And astonishingly, even a disease like schizophrenia, which is just, baffling. There's a researcher, a psychiatrist researcher in Germany named Carsten Korth, who has demonstrated prion-like activity of a protein called DISC1, which is implicated in schizophrenia. So it's a brave new world. Some of it seems crazy, but there's a lot of utility in the concept.
You mentioned that the antibodies could prolong survival in about 25% in the ALS. As far as quality of life and symptoms, did that show any benefit?
This was a mouse experiment.
Oh, I'm sorry.
I guess the answer is it's hard to tell.
Okay, thank you.
But I'm saying that because generally ALS kills people in 3 years, within 3 years. So if we were able to get a similar prolongation, it would be a year or so.
I asked because I have a friend whose father passed within the last year, and he was a 20-some-year survivor of ALS.
Yep. That happens too. You needn't look any further than Stephen Hawking. And talk about being cognitively intact.
Okay, I have a question for you. I'm over here. Back here.
Where is he?
Over here.
Okay.
Yeah.
Sorry.
So with that, with the thinking that the schizophrenia, diabetes, those other diseases may also be prion diseases, if there is a family history of any of those, does that maybe put you at more of a higher incidence of having or developing the CJD? Is there any association or correlation between family history?
So certainly there's a correlation between family history and prion diseases, and there's a correlation between family history and schizophrenia. But that, these diseases are genetically inherited. Familial prion disease with a mutation in the prion protein gene, and schizophrenia with mutation in DISC1 or any one of several dozen candidate genes can put you at greater risk for schizophrenia. Okay. I am told last question, so very appropriately, Debbie. Yeah.
These linkages that you're showing, which I know you've been developing for many years, has this increased your ability to raise funds?
That's a very interesting question, and one would think that, if the goal of the lab, my lab, is translation, that we'd get a lot of interest from the funding agencies. But it's not the case, or at least in Canada, it's not the case. So I struggle for funding, and I get grants from disease-related organizations and from donations, and also industrial contracts. But it's hard getting that kind of money.
We're going to
Okay.
go to break now.
Okay.
Thank you, Dr. Cashman. Thank you.